Hi, my name is Sarah Esstman, and I'm an associate professor at Wake Forest University in the Department of Biology. My research program focuses on viruses. We're fascinated with how viruses replicate. I think right now the world is fascinated with how viruses replicate. And for several decades now, we've been keenly interested in mechanisms of viral replication inside of the cell. The virus that we study is a virus called rotavirus, which is an important cause of gastrointestinal disease in many animal species, including human children. The burden of rotavirus disease is very severe, with estimates of over 100,000 children dying each year from infections. Right now we can see an image of the rotavirus virion particle, and we can see the rotavirus genome, which is the instruction manual for the virus. The questions we have about rotavirus are related to how is this genome, or the instruction manual, replicated inside of the cell. And the principal viral protein that mediates this process is a protein called an RNA-dependent RNA polymerase. And we study the polymerase using various structural and functional approaches. Our primary approaches that we use are what we call structure-based biochemistry. So we do quite a bit of structure-guided mutagenesis and in vitro assays. And then we also have questions that are related to how our rotavirus is evolving during their spread in human and animal populations. Specifically, an interesting feature of rotavirus is that its genome, or this instruction manual, is made up of multiple different segments. So we can see those segments here. You can actually kind of separate them biochemically in something called a gel. And we're interested in how these segments might be swapped among strains that co-infect host organisms or host cells. This process of swapping is a process called genome segment reassortment, and it's one of the major mechanisms of creating genetic diversity for viruses in nature. So we're fundamentally interested in what is this process doing, how is it driving strain evolution, and what are some restrictions on that process. And for this project, we also, in addition to doing biochemical type assays, we also use quite a bit of molecular genetics as well as bioinformatic approaches to tackle these exciting questions. Now let's go meet my lab.
Hi everyone, my name is Mackenzie Brown and I'm a PhD candidate in Dr. Sarah Esstman's lab. The Esstman lab is widely focused on protein-protein interactions that allow for rotavirus replication. Rotavirus is a double-stranded RNA virus, and as we know from the current pandemic, RNA viruses such as SARS-CoV-2 has the ability to cause extreme health issues and economic defects. Specifically, rotavirus and other RNA viruses contain an RNA-dependent RNA polymerase, which is the polymerase that catalyzes important stages of RNA synthesis, such as replication. Our lab focuses on the regulatory partner of the polymerase, the core shell, and tries to use rotavirus as a model system to understand this RDRP regulation that occurs in multiple RNA viruses. Specifically, my project utilizes biochemical and structures of the rotavirus virion to inform mutants, BP2 proteins, the core shell protein, to then further understand how these mutants and their contacts regulate the activity of the polymerase and essentially rotavirus replication.
Hi, my name is Sarah Nichols. I'm a first-year PhD student in Dr. Sarah Esstman's laboratory. I came here from Maine, and as an undergrad, I studied microbiology, and I was really interested in infectious diseases and the burden of infectious diseases on our society as a whole. And when I came here and interviewed with Sarah, I found her work really fascinating and really interesting. I think that she has a really unique approach to solving viral pandemics and viral problems that occur throughout the world. And I find rotavirus really fascinating. I think Mackenzie talked to you guys a bit about her research and how we're focused on protein-protein interactions in this lab. But another avenue of research that we do is looking at rotavirus replication as a whole, and specifically where that occurs in the cell. And that's what my project is kind of starting to turn into. I'm looking at viral replication factories and the signaling that occurs to generate those factories inside the cell, and the signaling that traffics viral proteins through the rest of the cell in order for the virus to leave and infect other cells.